this lecture is on cancer. This is a PowerPoint from the National Cancer Institute. This is actually not the entire PowerPoint that you would find on their website. It is just the slides that I felt like were most critical for what we're learning. So cancer is related to what we're talking about because it's really just a matter of a loss of normal cell division. So you can see in this diagram that in normal cell division, if for some reason the cell is damaged, then it'll either be repaired, or if not, then the cell will die through apoptosis. In cancer, what happens is when a mutation happens, an error in the DNA, that cell doesn't stop dividing, and it acquires another mutation, and it's still dividing, and it acquires more, and it's still dividing, and now it's getting even more mutations. And now it's making a mass of cells that are extremely mutated and very far from what they should look like or how they should function. And so this results in a cancerous tumor. So this is an example of normal cell growth in the skin. This is the epidermis, okay, outer layer of the skin. And as you lose cells or shed cells from the top layer, cells continue to migrate up. And so the bottom layer becomes the top layer, and then you've got a bunch of dividing cells in what we call the basal layer here. And then below that is the dermis. And so these cells just continue to divide, and then as they divide, they are older and older and older, and they migrate up, and then the oldest cells shed off. So what happens here is if you have sorry, I had to break for a second. This shows, say this cell here becomes abnormal and does not go through apoptosis like it should, and it continues to divide, it will actually not only divide and create cells over here that are all infected, it will actually send signals or cell communication to other cells in the neighboring area and cause them to be mutated as well. And then they'll divide and they'll divide all those area cells until eventually you've got this whole huge mass of cells that are not normal. They're somehow damaged and don't function properly. So this can be bad, right, if you have a tumor that is cancerous. However, when these cancer cells start to invade other tissues and they enter the bloodstream, this is really dangerous because once they enter the bloodstream, they can travel to another part of the body and build another tumor there. So this is the skin. So these cells can get into the bloodstream and maybe they travel to the liver. And then all of a sudden you now have a liver tumor. And then maybe they travel to the brain and now you have a brain tumor. And so this can be a really, really difficult thing because it's like you're constantly chasing a moving vehicle. They keep going to other and new locations and growing new tumors there. So this is really hard to treat because you can't just surgically remove one tumor because they're going to pop up in other areas as well. We call that metastasis. When it metastasizes, that means the cancer cells have become bad and they have gotten into either the lymph system or the circulatory system and they're invading other tissues. And this just shows a benign tumor, the difference between the two. The benign tumor is not a cancerous tumor. These cells have divided uncontrollably, so there's more cells than there should be. See, there should be like one line here, and there's multiple, but they're not malformed. They're not dysfunctional. They're not bad. They grow locally, and they cannot spread into the blood and metastasize. So that's what makes benign different than malignant. Malignant are actually your cancer cells. They're going to invade neighboring tissues. So they're going to recruit other cells to be bad with them. They're going to enter the bloodstream, and they're going to metastasize into different sites, into any other organ. And if you see this down here, this is also important, that over time, a benign tumor left untreated or not surgically removed could become a malignant tumor if you just add more mutations. So the DNA mutates and more mutations. I shouldn't talk and write at the same time. All right, so usually what happens is if a doctor finds a tumor of some sort or a mass of cells, so they find a skin mole or something that looks a little abnormal, they'll do what's called a biopsy. Excuse me. And in a biopsy, they're going to take the tissue. Sometimes they'll do a blood sample for a biopsy too, if it's a leukemia or something. And then they're going to do some pathology on it. So they're going to actually take it and look at it under the microscope. And what they're looking for are these ones. These guys are bad because these are bad cells. These are obviously misformed, malformed, I guess you could say, and they're not going to operate properly. And then there's also some things they can do genetically to look at the genes. And they've done this with breast cancer. They've actually looked at some of the genes and identified genes that are associated with breast cancer. They just take your blood and then do a genetic profile on the person. So looking at the microscopic appearance of cancer cells, when you look at them under the microscope, normal cells are round, they're circular, they might be, kind of cuboidal shape if they're skin cells, but they have a normal shape. Cancer cells are going to be irregularly shaped. And then in a normal cancer cell, the nuclei is kind of a normal size, takes up a lot of space, but it's kind of have a defined circular shape. And cancer cells are large, and then the nucleus is not necessarily circular. And then compared to the nucleus to the cytoplasm, there's not as much cytoplasm volume relative to nuclei in cancer cells, and they can vary in size, shape, and just abnormal structure. They can also have a loss of normal specialized cell structures. This would be like, the dendrites on a neuron, and these would not be normally shaped. They might be disorganized or not arranged neatly, and they have poorly defined boundaries and identifying where one cell starts and another begins. This is normal skin cells or normal cell replacement. Could be something besides skin, but it's how cells are replaced. This group goes away, and then these are recruited up, and then new ones are formed below that layer. Hyperplasia is not necessarily cancer. It's just you have extra growth here. You have more cells than you should. Dysplasia is not necessarily just more cells, but it's cells that are abnormal. You can see the difference in how these cells are organized and shaped. This can be a problem. When you talk about cervical cancer, this is a cancer you can find in the cervix of a female. Every year, women are required, or not required, I guess, but are recommended to have a pap smear. And in that pap smear, one of the things they're looking for dysplasia, abnormal cells. They might have extra cells, that's not as big of a deal, is if they see cells that are more, but they're also irregular. So what actually causes cancer? Well, that's a good question. There's a lot of things that can lead to cancer. And scientists are newly exploring and looking at different things all the time. It could be your genetics. You could have inherited genes that make it more likely for you to get cancer. We'll talk about some of those soon. Some viruses can lead to cancer. One of the ones that I can think of that is really common is HPV, and that's the one that leads to cervical cancer. When I was a kid, they didn't have a vaccine for this, but they now have a vaccine for you teenagers to get. It is a sexually transmitted disease. It's a virus, but it leads to cancer in females on their cervix. Some bacteria too. I can't think of a good example of that though. Chemicals, think of like asbestos, is a big one that many years ago they used to use as a fire retardant that now is known to be a carcinogen. A lot of chemicals that I even we order in like a chemistry lab, you can check, and some of them will say that it's it's a carcinogen, and so you shouldn't be inhaling it or allowing your students to drink it. Radiation, best example of this, UV. X-rays also are a form of radiation, which is why you should limit the number of X-rays you have. Usually doctors won't do those unless they deem them really important and appropriate at the time, just because the more X-ray exposure you have, the more likely you are to damage your DNA, and that could lead to cancer. So this is just a graph kind of explaining the incidence of cancer. So cancer incidence low versus high on the side of the graph. And then annual sunshine, the least amount, like in Detroit, they don't have as much sunshine as Dallas does. Dallas has a lot of sunshine, direct sunshine. And you can see the incidence of cancer is much higher in Dallas than it is in Detroit. Cancer risk and aging, the number of cancer cases per 100,000 people. Age of the person, you can obviously see colon, breast cancer, the amount is much, much higher the older you are. Well, one of the reasons for that is one of the causes of cancer is DNA mutations. And we get mutations to our DNA on a very regular basis. Think about skin cancer. You're exposed to, every time you go outside and you're exposed to UV radiation, it's damaging your DNA. Well, the more damage you get to it, or the more accumulation of damage, the more likely it is you're going to get cancer. Well, somebody that's lived 80 years has acquired a whole lot more damage than somebody that has, only lived 10 years. So that's one of the reasons that with most cancers, the incidence goes up the older you get. And then we'll talk a little about genes in cancer. So, these are all environmental things. Chemicals, viruses, radiations, things that are in the environment, we may or may not be able to control those. Heredity is something you can't control. This is your genetics. This is what you're born with. And scientists are getting more closer and closer to identifying more and more genes that predispose people to cancer. So DNA structure, just kind of a brief overview. We learned about this after Christmas break. But if you remember, it's a double helix, and it's twisted. So a double helix means it's a helix that's like a staircase twisted, and it's double, meaning that there's one strand here and one strand here. And there are bases in it, A, T, G, and C. A stands for adenine, thymine, cytosine, and guanine. And A and T are always paired up, and C and G are always paired up. Well, sometimes they're not, and they're paired incorrectly, and that can be an example of an error or mutation. So if this is your normal gene, okay, that's coding for something, and there is a single base change, so instead of having a C and a T, there's a C and a G, that can be an error or a mutation. A single base change one way, addition, meaning you're adding something. So notice there was a C and a G here, but we've added an extra G in there. And also taking things out, so we've removed the C and a T. These are just different types of ways that DNA can be mutated or changed. And anytime a gene is changed, it can affect the outcome or what the individual experiences. And in some cases, that means they may experience a greater risk of cancer. So there's a couple types of genes that are of concern. One of them, one group is called oncogenes. So in a normal cell, your genes are going to regulate the cell growth, the cell cycle. Okay, they're going to make sure that cells divide when they should and die when they should. Oncogenes are genes that are going to accelerate cell growth and cell division. They basically are going to keep that division going when it shouldn't go. So if you have a mutated oncogene, that's going to mean that the cell growth cycle is going to be affected somehow. It's not going to grow when it should or not grow when it shouldn't. Okay, if you remember, I said a bit ago that DNA can be damaged. Well, a proto-oncogene is a gene before it becomes damaged. A proto-oncogene can become an oncogene. Oncogene is a damaged gene. Okay? And you don't want to have these. Not want oncogenes. These oncogenes stimulate cell growth when you don't want the cell to divide and grow more. So, for example, when a growth factor reaches a receptor on a cell, when we've talked about cell communication, growth factor reaches this receptor, produces signaling enzymes, and then that turns on transcription factors that are going to make a gene. Okay, it's going to make that gene active. It's going to make the gene, and it's going to produce protein. Okay, and when it does that, cells are going to divide. You're going to get lots of cells because it's turning on genes that affect cell growth. Well, you don't want this if you want the cell to die. You don't want it to continue to divide and divide and divide and divide and divide and then not stop. So oncogenes are genes you don't want because they are damaged and they produce more cells than what you want. They have an abnormal amount of growth. Okay, so this is what normal growth is happening. But oncogenes create lots of growth, extra growth that should not be happening. So there's extra transcription here that shouldn't be happening. That's what proliferation here means. Okay, another group of genes related to cancer are tumor suppressor genes. These are genes you do want. Okay, you want to have tumor suppressor genes because, like their name suggests, they suppress tumors. They suppress cell growth. If we were to kind of compare tumor suppressor genes to oncogenes, oncogenes are like normal cell growth as you put your foot on a gas pedal and an automobile, and you're just going, and you can release it and keep going and press down harder, and you have control of that. An oncogene is like a gas pedal that is perpetually on. It doesn't stop, and so you're making extra cells you shouldn't make. In contrast, a tumor suppressor gene is like a brake pedal. It tells the cells to stop growing and stop dividing, and you want that. You want the brake pedal. If you don't have a brake pedal, that's a bad deal, okay? And so what happens here is, for some reason, you can genetically inherit a tumor repressor gene that doesn't exist. Either you don't have it or it's not working, it's inactive. And what that causes is cells to not stop dividing when they're supposed to. And you can get cancer cells from that. This is that analogy with the brake pedal. One of the known tumor suppressor genes that is very widely known to affect cancer is the p53 gene. They found this in a lot of different types of cancers. They've sequenced DNA of people with cancer, and they find this one very, very commonly. And this gene triggers cell suicide. It triggers what we call apoptosis. And so a lot of people that have cancer lack that particular gene, the p53 gene. This is what's supposed to happen. You have a normal cell, excessive cell damage, cell dies. Well, if you don't have p53, that cell is going to continue to divide. And then there's a lot of DNA repair genes that they're identifying that basically when you get a DNA that is damaged, it will repair that damage, and then you get no cancer, and it's not a big deal. But sometimes when you have like this A is not, or T is not supposed to pair with C, it's supposed to pair with A, you get this, it would correct it, no cancer. If it doesn't correct it, then we can actually get cells that continue to divide. And so they've looked at several DNA repair genes being non-functional or ineffective in cancer patients. Cancer cells tend to involve multiple mutations. It's not like you can acquire one mutation and all of a sudden you've got cancer. Usually a cancer cell, as it starts from excessive mutations. Mutations to oncogenes, or proto-oncogenes, causing them to be oncogenes. Mutations to tumor suppressor genes that then are not working. Mutations to DNA repair genes. So it's a multiplication or a lot of different types of damage that can happen that will usually lead to cancer. It's not just one thing. Genes that are implicated in cancer, we talked a little bit about these oncogenes, tumor suppressor genes, and the DNA repair genes. These are the three main ones that they've identified. But other mutations can also occur in cell death genes, cell signaling. One cell is not sending a signal to another. The checkpoints in the cell cycle that we talked about. All kinds of things. Cells that are a part of differentiation process, which is how your cells become specialized. And then carcinogens can actually activate or deactivate genes too. One thing that's really unique about cancer cells compared to any other cells that are dividing is that cancer cells tend to corrupt the surrounding environment. They recruit a blood supply. They send signals to other cells in the area and tell them to be bad. These are like the really bad kids in school. Not only are they bad themselves, but they can somehow find others to go be bad with, and those people will be bad too. So these are the bullies. These are the head honchos of the gangs. They're the bad guys. And that's it for this.